A brain cancer lab at UCSF says it cut analysis time by 90%. Same AI model everyone already has, a different bench underneath. Stephen Francis studies glioma, a brain tumor, at the UCSF Brain Tumor Center. His own estimate, Claude Science, cut his analysis time by about 90%. He calls it the first truly agentic AI made for his field. The twist: this is not a new model. Claude Science is an app, a workbench running Claude models that already shipped. By the end, you'll know which results are real, what the bench adds, and the 4 places it fails. Start with the scoreboard. At the Allen Institute, neuroscientist Jerome Lecoq wired 20 custom skills into a review template and produced over 10 literature reviews, each above 100 pages, citations AI verified. Work he estimated at 2 years took weeks. One team reported it flagged the lab virus contaminating their RNA data, unexplained for nearly a year. Another reported a germline analysis in a tenth of the usual time. And Manifold Bio, a drug discovery company, screens millions of protein binders for tissue-targeting drug candidates. So how does an app pull these numbers from a model that didn't change? Because the bottleneck was never reasoning. It was the maze scientists juggle: databases, file formats, notebooks, terminals, cluster queues. Claude Science collapses them into one environment on Mac or Linux, or a remote cluster over SSH. Inside sits a coordinating agent with over 60 pre-built skills for genomics, proteomics, and drug chemistry, plus live connections into the major biology databases and NVIDIA's BioNeMo protein models. It renders 3D protein structures and genome tracks natively and writes figures and manuscripts next to the code. Labs save their own pipelines as reusable skills, exactly how Lecoq got his 20. Now the part that makes it publishable. Speed is worthless in science if you can't defend the result. Every output carries its code, its environment, and the history that produced it. A built-in reviewer agent hunts citation errors, numbers that trace to nothing, and code versus figure mismatches before a human opens the draft. Kelvin Lau at EPFL praises exactly that: the step, the code, the why. And it runs on the lab's own machines, so raw patient data never leaves. Only the context each step needs goes out. Lecoq's framing is the tell: not just faster, far more information about provenance. The pattern reaches beyond this app. In Anthropic's separate protein binder research, Claude hit 14 of 15 targets, a 22 to 35% design hit rate versus the usual 10 to 15. Stanford's Biomni, a separate Claude-based agent, ran a genome-wide association study in 20 minutes. At MIT's Whitehead Institute, a CRISPR screen system caught patterns its own lead scientist missed. Same lesson: the model was ready, the harness was missing. Now the 4 limits, stated flat. Reviews still contain mistakes and need a human check. Paywalled papers are out of reach. Biosecurity guardrails block some legitimate queries. And it can't judge whether a paper is any good. The 90% and the 2 years in weeks are researchers' own estimates. But none of it came from a smarter model. It came from a bench where every number is traceable. The open worry: a flood of fast, thin, AI-written science. It's live on paid Claude plans at claude.com/science. If you want what AI has verifiably done, not promised, subscribe.